Dr. Claudio Parolo, who will talk about engineering electrochemical Aptamer-based sensor from optical characterization to implementation onto gold electrodes. Dr. Claudio Parolo is an associate researcher at the University of Barcelona, specializing in rapid diagnostic tests for infectious diseases. His expertise spans electrochemical Aptamer-based sensors and lateral flow assays supported by extensive research at institutions such as University College London and UC Santa Barbara. So Dr. Parolo's innovative work has advanced biosensing platforms for healthcare and travel medicine. Dr. Parolo, we look forward to your presentation. The floor is yours.
Thank you. First of all, thank you for the introduction and the invitation to this very nice event and super well organized also. So yeah, first of all, I'm gonna go. Okay, first of all, let's see how we're gonna move the slides. What do I press here? Okay, now. Okay, so I'm gonna give a brief introduction to myself, especially for students here. At least when I was a student, I was also curious to know who the speaker was coming from and their professional journey. So originally I'm from Italy, from the north side of Italy. And I studied biotechnology in the University of Padova. And then during my master, I did my Erasmus period in Professor Mercosi group. And you see Barcelona by night, because as an Erasmus, you basically party a lot as well. So it was a good work hard, party harder. And then I stayed for the PhD students, and I got the opportunity to go to Harvard to Professor Whiteside's group for 6 months. Staying there was really opening my professional experience, because just by being there, I was invited for my first postdoctoral position in London at UCL. Basically, during all these years, I was working on paper-based platform, both optical and electrochemical, for infectious diseases. And in London was pretty much the same, but for early warning sensing systems. So we were combining like also data from internet to rapid diagnostic tests. And then I wanted to change a little bit also the weather. So I got a fellowship to go to Santa Barbara in Professor Plasko group. I spent there 3 years. And there I learned about electrochemical Aptamer-based sensor, which we're going to talk about today. Then I got the first kid. We needed grandparents nearby. I was lucky enough to get a fellowship to go back to Barcelona. My wife is from there, and you see Barcelona by day now because we don't party anymore. And then after 3 years, after 2 years in Professor Mercosi group again, I wanted to go closer to the application field. And so I joined, I got the second kid. And then I got to the Barcelona Institute for Global Health, which is a research institute that combines 2 hospitals, 2 universities, and we have also some research centers in Africa and South America. And then after 3 years there, I'm still working with them. I got a tenure track position at the University Rovira Virgili. And so basically, I have basically 2 groups now, and the idea is to go from the clinical needs at the hospitals to the chemical engineering department at the university, and then go back to the hospital to the validation. So moving on the real talk. So after the nice introduction of VASA, I'm going to focus a little bit more on in vitro diagnostic, which is basically techniques that use biological fluid to assess the presence of a specific medical condition. No? So we saw already like the COVID-19 rapid test, but in vitro diagnostic, they're also included, for example, PCR. No? And you can see there are very different techniques. But in both cases, this is a very huge expanding market, as you already know. So I think we are well positioned for the future. And focusing on rapid tests, there is another definition that is always used, that is point of care testing. No? So it's basically a test that can be used near patients when it's needed. No? But it's still a general definition, because point of care could be, when we think about it, we probably think of something like this in the most case scenario, like some very poor funded, let's say, or very complex situation. But it's also could be something like this. So it very like, for example, an isolation room in one of the best healthcare systems in the world. So if we go a little bit deep into these scenarios, we can see something that struck to your mind. So the first of all, what you see is that there is no electricity, right? Or at least there seems to be no electricity. So we cannot really rely on bulky equipment that require a lot of energy. At the same time, we don't have a cold chain probably, or it's going to be very efficient. So you cannot really rely on reagents that are very delicate, for example. There are probably few hospitals in rural areas, so people are not maybe go once and then they don't come back after a few days. So you really need something that must be very quick, and probably you're gonna have very few trained personnel there. In between the two, we have a second options that is like when you go to your doctor office, when you're like flu, you go to your doctor, you don't go to the major hospital. And there is a work, and I forgot to put the reference, but basically they established the average time for your doctor visits, and they found that, for example, in Bangladesh, there were 48 seconds, and in Sweden is 22 minutes. So although they are very different in minute scale, but it's still not very much. So, and in this case, so if you want to develop a sensor that can be applied here, you still need something that is pretty quick. Also, your doctor probably don't have very huge resources, so you probably have a small office or may come to your house. And the doctor also needs to probably assess very different symptoms. No, you can go with fever, but you can go with cough, you can go with very different things. So you need something very little that can be applied to very different diseases. And also probably your doctor is not as trained to molecular biology, so it really needs to be something really easy to use. And finally, we have the other end of the spectrum that we saw before, which is like basically emergency room or intensive care units in tertiary hospitals, so the very big one hospitals. And in this case, you have different type of needs. No, of course, you see like, you see there are a lot of sockets, you have a lot of equipment. But in that case, you still need urgency. No, you probably the patient that arrives there probably need very quick interventions. They need continuous monitoring to be sure that they don't the symptoms doesn't go too severe. And then in any case, there are still not infinite resources, so there are limited beds, for example. So you really want to keep the patient in the hospital the less as possible. At least that's what the philosophy of healthcare systems are, no? So if you can combine all of these three scenarios, there is a nice work from Land. The reference is the next presentation. You'll probably be aware of this work, is the reassured criteria. Basically, they divided the different environment where you can apply a point of care test into this pyramid. Can you move around? Yeah, I mean, so on the top you basically have like the very top hospitals, or maybe I don't know.
Pointer.
Okay.
So generally, if you press here.
Okay. Thank you. Okay, so basically at the top you have a national specialist, that is basically the intensive care units. No, so in this case, what they say is that the test that should be developed for this type of environments, they should have the highest accuracy ever, but we can avoid to be. I mean, they could be a little bit more expensive and a little bit less easy to use. Let's say, no, because you're already in a very specialized environments. Then in the middle one, what we said with the doctor office, probably something between 1 and 3. You still want quite high accuracy, but you still want to have more accessibility and affordability, no? Because you're gonna deploy it to many different patients, and so you still want you really need to improve the accessibility. And finally, the last one. So let's say the real point of care, rural area. You really don't, it's not that you don't care, but you cannot accept less accuracy, but you really need to have your sensor accessible and affordable, because even though it's not super accurate, the possibility to deploy to very different people is still providing a very advantage to their health. And so on this work from Land et al. It's called Reassured Diagnostic. Is basically they define a series of criteria that define what is the ideal test for infectious diseases specifically. So they need real-time connectivity. So you basically need the test to be connected to your doctor, but also to the healthcare system, for example, to identify new pandemics. No, Should have easy specimen collection, so you don't really need centrifugation or complex systems. You just want, for example, saliva or a finger prick of blood. Should be cheap, sensitive and specific. Of course, you want to diagnose correctly. That should be user-friendly, as we saw. In all cases, you want something to be really easy to use and rapid and robust, no? And the other two is that should be equipment-free or almost equipment-free. So something very little, and shouldn't be produced too much environmental pollution, for example. No, you don't want something including some toxic reagents, for example. And finally, should be deliverable to the end user. So this is taking this as the ideal characteristic for a biosensor for diagnostic applications. We are working on this is so-called electrochemical Aptamer-based sensors. I saw that you have already been working a little bit of that, so apologies if you already know some of these parts, but maybe you have some question that I can answer. So the one that we work on, because you can use Aptamer in very different ways. Now, the one that we use is based on use of gold electrodes. Most of the cases, you have a monolayer that attaches your aptamer through a thiol gold bond, and then Andrew will talk more about the formation of thiol interactions. Then you have your aptamer. For those that are not aware, basically nucleic acid sequence that specifically recognize not nucleic acid targets. And then at the end, it's modified with a redox reporter. In this specific cartoon, you see how the redox reporter is actually kind of far away from the electrodes. But then what we do is after adding the metabolites, you have a conformational change, which changes the structure of your aptamer. In a way, it's something related to the previous talk on mechanics. And so it makes the methylene blue, the redox reporter that you're going to use closer to the electrode surface. This in turn generates a different electrochemical signal where in the absence of your target, you have a lower signal because your reporter is far away, so you have a less efficient electron transfer. And when it goes closer, you improve your signal. And the difference in the signal can be used to quantify the amount of target that you have. So why this is a reassured sensors? Well, first of all, because it can provide real-time connectivity, can be attached to a smartphone. And it also allows for continuous monitoring, as we'll show some example at the end of the talk. It has ease of specimen collection, yes, because we show that we can really use it in a very wide variety of samples from whole blood, urine, saliva, you name it, we've pretty much done everywhere. It's affordable, it's like a glucometer, as we saw before. It's still one of the most widely employed rapid tests. Sensitive, well, sensitive really depends on the application. In some cases, if you see on the overview of our sensor, we don't have any amplification steps. So the sensitivity of our test really depends on the aptamer that we selected, but also on the requirement of the application. So in some cases, we don't really reach the required limit of detection. But we definitely can have specific results. We'll show very briefly, since you're familiar with aptamer. When we select the aptamer, we can include like counter selection strategies. User-friendly, it's basically doesn't require any step. You just put the sample and you press. So you can both quantify at the point of care, but you can also use continuous monitoring. Same thing, so it's basically you can get results in seconds. It's not really equipment free, as we saw for the glucometer, but it doesn't really require very bulky equipment. So it can be, for example, powered with your smartphone if necessary. And it's environmentally friendly because you can use very small electrode chips, and you can use like also environmentally friendly substrates. And finally, yes, can be delivered to the end user. And since it relies on aptamers, it's also much more resistant to, for example, different temperature conditions. So it can really be shipped anywhere in the world. So during this presentation, I'm gonna, let's say, support the protocol that I'm gonna explain with data that we have selected in the last few months. It's not published yet, so it's sort of confidential. But basically, we are trying to work, as I said before, with ISGlobal and working with the tropical medicine unit. And and especially in this work, we're working with imported malaria. So here you can see you have the years and you have the numbers of international travelers. You can see how the international travelers increased every year. This is what the SARS epidemic, financial crisis, boom, COVID. But in just 2 years, we already take the trend. And actually, we are over, sorry, 1.5 billion travelers this year. On those 1.5 billion travelers, 60% goes to tropical areas where there can be endemic diseases like, for example, malaria that we're working on today. And the problem with malaria is that it has at the beginning like very random, very general symptoms like fever. But in imported malaria, so international travelers, meaning adults that have been raised in non-malaria endemic country, are the third population most at risk after children and pregnant women. And of those, basically 12% get severe symptoms that can go from cerebral malaria, shock, respiratory distress, kidney injury, hyperparasitemia. I mean, it can be very tough. And 5% of them actually die from malaria. So in this context, I'm working with 3 medical doctors led by Professor Munoz at the International Traveler Services. And we have also an app if you want to download. If you plan to travel in any of these countries, you can download it, and you have access to our medical team 24 hours a day. And they speak perfect English, so it's a very good service if you ever plan to travel in some of these countries. So specifically related to malaria diagnostics, why we're working on a new test is because currently the gold standard are laboratory-based tests. So basically, most of them are PCR and microscopy. And of course, as we already saw, both are not really point of care because you need either a bulky equipment and some say specialized personnel, and the microscopy also need a really specialized personnel to really understand, to really see the parasite inside the red blood cells. And in both cases, it takes several hours to days to get the results. The alternative is rapid diagnostic tests like lateral flows. And in endemic countries, they actually work pretty well. They have very good sensitivity and specificity. Unfortunately, they're not really implemented in non-endemic country. That's one of the problems. And the other problem is that the parasite is actually developing and is actually deleting the gene expressing the main antigen. So we are seeing a lot of false negative results. So in this context, what we want to do is integrate in devices like this and electrochemical Aptamer-based sensors to be used pretty much everywhere. And I already thank her because all of the data are done by our postdoc, Chuyue. She's a machine. I'm super happy to have her in the group. Okay, so let's move on more on the practical aspects. As we already saw the main components, we have an electrodes. So to work with an electrodes, we need a potentiostat. What I suggest is using a multi-channel potentiostat during the optimization phase, because it can be a little bit tedious. Otherwise, it can take several hours to optimize it. Of course, when you move to the application, you probably want something small like this. And regarding electrodes, during the optimization phase, we use these gold rod electrodes, because we can really polish them, reuse them many times, and they provide very reproducible results. And then once you want to prove that your sensor works, you can move to this type of electrodes, although they are much more trickier to work with for optimization experiments. So everyone here knows about aptamers? Is anyone that doesn't know about aptamers? Okay. Okay. So basically, aptamers are, as we said, are nucleic acid probes that binds not nucleic acid targets. And they're generally selected by a process called SELEX, which stands for Systematic Evolution of Ligands by Exponential Enrichment. So you basically start by buying a library of nucleic acids, which have a forward primer that is fixed, a reverse primer that is fixed, and then you have a random library. So you basically have very random sequences here that allows you to have 10 to the 15 to the 16 individual sequences. So you have a bunch of different sequences of nucleic acids. Then in the normal SELEX, you have a magnetic bits modified with your target of interest. and you incubate your library with your target, basically on your bits. That by magnetic separation, you basically have just the aptamers that stick to your target are kept. And what you go then, you amplify the one that you want, and you basically have an enriching in the aptamer sequences that are specific for your target. The other thing that is crucial is that we can do counter selection strategies. So once you have enriched some of those sequences, you can challenge them with the potential interference. That could be similar proteins or other, like, for example, human serum albumin. And then in this case, instead of amplifying those that binds, you amplify those that do not bind. And then in the end, you end up with a sequence of nucleic acid that specifically recognize your target. The problem with these methods, although sometimes it works for our application as well, is that it doesn't ensure the conformational change upon binding. So what we recommend is try to either do yourself the capture selects approach, which is basically, it's a little bit different from here. In the capture selects, you have your, let's say, library is attached to the magnetic bits. And the presence of the targets is basically displacing your aptamers from the magnetic bits. And to be displaced means that they must undergo a conformational change. So aptamers selected with the capture selects are probably more, let's say, probable to work in our technology. Although it's a little bit more complex and more expensive strategies, it's probably sometimes worth to give it a go. And that's basically what I was saying, is that once you're selecting an aptamer, they generally achieve a 3D structures. And the binding doesn't mean, in this cartoon, it's very simplistic, but if your single strand sequence is like this, then once it hybridize, it can bind the target. But since our sensor rely on the difference between these two positions, if it doesn't really affect them, we would have a no signal response. So what we do to induce that conformational change, we rely on software such as NewPack, and then Mariana can talk more about it. But basically, it's a software where you can put all the different information, if it's RNA, DNA, temperature, etc., and you end up with a potential structure of your aptamer sequence based on your temperature. Like in this case, you see we have a stem loop here of 5 bases, and we have some sorts of loops around here that are probably involved in the binding itself. So what we did for malaria, we didn't do the selects ourselves because there were a lot of aptamer already published. In this specific case, we took an aptamer published in this journal by Limson Group. But the aptamer was pretty long, was 46 bases. So one thing that you can do is start cutting it from both sides. So basically, and this is what we achieved. So this is the, let's say, parent sequence, the original sequence. So you can see again here, there is a stem here, some loops around here. And what we did was starting cutting from this side. First, we cut 6 bases, and then another 5, and then another 5, and then also on that side. And you can see P1130, we have already like completely destroyed these loops at the beginning, P1135 almost. So once you have selected your aptamers and you selected your ideal potential structures, what we do to make everything quicker and do not spend too much money is do fluorescent analysis before moving to the electrochemical methods. Because electrochemical methods, as I said before, we use like these big electrodes, and you cannot really multiplex as much as, for example, with a 384 plate, where you can do many different calibration curves, and you can use very limited amount of samples compared to this. Like in this case, we generally use between 2 and 3 milliliters each time, and here you can use 50 or even less microliters. So it's much more efficient. And so what we do, we basically purchase our aptamer sequence with a quencher and a fluorophore. In this specific case, the fluorophore is away from the quencher, so you have a nice fluorescence. When it changes conformation, the fluorophore can get closer to the quencher and the fluorescence decrease. This is just an example, can be also the other way around depending on the aptamer sequence. In our case, we purchased the aptamers with the fluorophore on their 5 ends. It's a 6 FAM, and on the 3 end, we purchased a quencher which is called Black MN Q535. And we purchased our target, our aptamers generally from Biomers. I don't know if you're familiar with the company, but it's a European-based company. It's very quick. They are very quick. They provide pretty good quality. And what is amazing is that you can have huge amount of different modifications. You can see like on the 5 prime, you can get, look how many different modifications. And specifically, so also for electrochemical, of course, and specifically for fluorescent measurements, they have a nice combination of quenchers and the respective fluorophores. Like in this case, we took the FAM and we took this fluorophore over here. And these are the results of the fluorescent optimization that we did. So you can see here the 6 aptamers that we tested, the original one and the different truncations. And then we tried with, so one thing that you should always keep in mind is always trying different aptamers concentrations. Otherwise, you can get to an excess of aptamer or not enough aptamers to have a nice signal. So these were the concentration of aptamers, not of the target. So we tried 10, 30, and 100 nanomolars. And then we added 500 nanomolar of LDH, 300 nanomolar of LDH. That is our target. And you can see how these 3 aptamers were really responding very well. So here we plot the signal to noise ratio. So we basically divided the original signal before the addition of the aptamer with the signal we obtained after adding the aptamer. And you can see how P1135, but also P1130 and P1140 were those that were responding better during this optimization phase. So after doing this preliminary test, then we moved keeping 30 nanomolar, as was the concentration of aptamer giving the higher signal, and we did the calibration curves. And you can see how the red ones, that it was P1135, was the one giving the highest signal to noise ratio, although also the other were responding pretty well, and they could be fit to a 4-parameter logistic curves. Okay, so we basically screen from 6, we moved to 3 aptamers. We did also some kinetic studies to be sure that the aptamers were binding pretty quickly. In all cases, they were binding in a matter of seconds to minutes. And the signal was also corresponding to the amount of target that we put. So you can see basically we first waited to be sure that the signal was stable, and then we spiked LDH, and you can see how the fluorescent measurements was changing. So we moved on to the optimization of the electrochemical sensors with those 3 aptamers. Again, I'm not going to talk about how to create the monolayer. Andrew's going to discuss more about it. But what is important here is that the signal of our type of sensor really depends on the frequency and the amplitude that you employ. No? It's not always the case, but if you are, let's say, lucky enough, you can get to a regime where you have signal off and signal on. And that's basically depend on the frequency and the amplitude that you employ. No? Like in this, so actually this work at the bottom explains really well how to optimize these 2 parameters and why those are important. But basically, here you can see the blue areas is where you get signal off, meaning that the original signal is higher than after the addition of your target, and the red one is signal on, meaning that the signal after the addition of the target increases. And so optimizing these 2 parameters, they show that, for example, using 25 millivolt at 20 hertz, you get a nice signal off response, the blue line, whereas 25 millivolts and 750 hertz, you have a signal on response. And what is nice, I'm not going to discuss much about it, but please let me know if you're interested. You can combine these 2 signals also to get a calibration-free measurement. So you can quantify the presence of your target just doing a calibration curve once, and then you always keep the same values, and you can apply those values to any type of target, and in one measurement you can quantify the presence of your target. In our cases, we fixed the amplitude at 25 millivolts, but you can see how, for example, the signal change for P1140, that was the longest one of the three selected, was very close to zero. So it wasn't really working very well on the surface. And P1135 was not even that good. It was around 10% signal change, but P1130 was the one more truncated, gave us a signal change in between 25 and 40% signal change. And you can see here how the signals looks like. So this is before the addition of the target. This is after the addition of the target for P1130. And this is how the signals looks like for P1135. So we moved on. We did the calibration curves. The calibration curve confirmed what we saw before. So P1135 has a calibration curve that goes up to 10% signal change, minus 10%, whereas P1130 was giving a very nice calibration curve with a very big signal change up to almost 40%. So we basically stick with P1130, and we did the kinetics also here. So you can see here the stability of our sensors. And then after adding 20 nanomolar of target and 200 nanomolar of target, you can see how quickly the sensor was responding to the addition of the target, meaning that we can employ also at the point of care in a very efficient way. The other thing that we did, of course, was the specificity test. In this case, we didn't select the aptamer ourselves, so we were not sure how specific was it, but basically this is the signal of our specific target. This is another protein present in the parasite at very high concentration, so it still responds a little bit, but not as much as here. And anyway, we were using concentration so high that is not even present in real biological conditions. And the last one is CRP, is a protein common in our blood. And you can see how the signal was basically not responsive. Unfortunately, it was not that good in 100% blood. We had to dilute to 1% blood. This is the stability over hours. So really what we are interested in, the stability in the first 15 minutes. So just to be honest, I'm going to show you that after a while, the signal starts to decrease. But you can see how the calibration curves exceed the decreasing of the signal. It fits very well. And again, the stability that you can see here is basically after 5 hours or 6 hours. But at the very beginning was super flat. So we could be employed also at the point of care. So some resources, if you are not familiar with them, one of the things is that if you start doing many of these measurements, especially in continuous monitoring, imagine that you do 10 hours of experiments and you do one measurement every 30 seconds, you have over 1,200 files for one electrode. So if you do a triplicate, you have much more than that. And since I'm not Neo and I'm not also a programmer, I asked the help of Sam. He is now a postdoc at Johns Hopkins. And he came up with an open source software that you can download. I'm going to show in the next slides, but basically does everything automatically. So as soon as your potentiostat makes the file, the program runs the fitting and extract the peak change, edit plots in real time. So in the supporting info of this work, we put some video tutorial, examples of data that you can use. And then you can see, you can use for square wave voltammetry measurements, CV measurements. And I think now they are working on an improved version of the software. And here is an example how it can work. So I don't know. He's actually talking. So if you download it, you will hear him explaining how things works. But basically you can see you have, you can add different parameters here, the durations, here the low frequency, the high frequency that you want to employ, start potential, final potential, and it shows you in real time how the peak appears. And you can basically tune, you can say, okay, I want to measure the peak between minus 0.2 to minus 0.4. So the software automatically fit everything and plot it. And you can see that then after a while, it starts to show the trend. Another resource that maybe you're familiar with is a Nature protocol wrote my supervisor at UCSB. It basically explains step by step how to do everything, from the polishing of the electrodes to the preparation of the tiles to really, it's a really well organized protocols that you can follow. And now I'm going to show you some examples of where we apply these EAB sensors. The first one is about the detection of kidney injury. So this is like the development of kidney injury over time. The current gold standard is detection of serum creatinine, but the serum creatinine can be measured after 48 hours, where the kidney is already being affected. What has been found that there is another biomarker called NGAL that can be detected in urine, and after just 2 hours from the initiation of the disease. So we basically created a fake bladder. Over here, we used a real urinary catheter. This is a real catheter that is employed in surgeries. We put the sensors inside the catheter. So you can see here, there are 2 sensors inside the channel of the catheter. We have a recirculating pump where we put urine and artificial urine. We use both. And here you have a pump that was adding NGAL directly into the beaker. And these are the results. So you see first the stability of our sensor for 5 hours. It was very flat. And then we added 2 nanomolar of target. You can start seeing a little spike, 15, 25, and 35. And these are all in the clinical range of NGAL. So here was the behavior after spiking the samples, and here the behavior by slowly infusing the samples with NGAL. So we basically started here, and after approximately 40 minutes, 50 minutes, we reached the 30 nanomolar measurements. You can see a nice steady increase, and then it was already stable again. Another example from the Mercosi group that we worked together, we basically tried, like everyone during COVID, to do something with COVID. So we wanted to do EAB sensors for COVID. So Andrea, who is now a professor at Tor Vergata, worked on the creation of this EAB sensor for the S protein. Here you can see the results after the addition of before adding the S protein was the black curve, and after the addition of the S protein. In this case, as we have a signal on, which if you can achieve it is much better because you basically avoid the doubts of having a decrease in the signal because the monolayer is destroying or not. And we show that it works in 100% bovine serum. You can see how it looks very nice for the calibration curves. And also in 50% artificial saliva, we were able to detect the target in a very efficient way. Another example that maybe you are not familiar or not, it's from Professor Arroyo. He was a postdoc with me in Plasco Group in Santa Barbara. And during his time there, he basically applied the EAB sensor directly in vivo. Well, this was not in vivo, but in other words, there was in vivo. So he put like the sensor inside the jugular vein of a rat, and through another vein, they were injecting different antibiotic and anti-cancer drug. And you can see how, for example, for doxorubicin, you can see the different spikes on the concentration and how the body was actually getting rid of the antibiotics over time. And this is for the demonstrated with doxorubicin, kanamycin, gentamicin, tobramycin, and is one of the most precise and time-sensitive technique that are currently available to monitor a drug, for example, but any other compounds, to be honest. Taking advantage of this ability to continuous monitoring something is that you can do the feedback loop control. So basically, if you can monitor in real time the presence of your target and you know that your therapeutic range is this one, for example, you can, as soon as, for example, your drugs go below the therapeutic range, you can infuse a little bit more to keep it constant so you avoid side effects and you optimize the treatment. And you can use it also, for example, in microfluidic devices. We publish a review. I forgot to put the reference, but basically what we were proposing is that if you can employ, for example, an EAB sensor in your lab-on-a-chip, you can automatize the way your lab-on-a-chip works. And as soon as you, for example, achieve a specific value of your target, you can, for example, tune the speed of your flow, you can stop your flow, or you can open some valves. So it's a really convenient method. So what we're doing now, I don't know, I'm going with the time, but it's basically the last. Okay. So the last one is what we're doing now. We have a project with a spin-off company from Arbenz Group. So we want to multiplex what we have been doing with LDH. So we want to detect both parasite biomarker for diagnostics. So we want to be able to tell the patient, yes, you have malaria, with host biomarkers like angiopoietin-2 and CRP. And we want to say to the patient, yes, you have malaria and you're probably going to get very sick in the next few hours. So you need to go to the ICU unit. Otherwise, maybe you have malaria, but all the other levels are fine. You can go home, take your pills, and you're fine. So it's a way to optimize the healthcare resources, but also for the patient itself. I mean, it's going to be good not to be in the hospital if you can avoid it. And we already done some biomarker discovery analysis. And in this case, we saw the association, for example, of angiopoietin-2 with severe malaria, which are these ones, compared to normal malaria. You can see we can discriminate very well between patients that got very sick from those that are not, and this is non-severe malaria. And so what we are trying to do now, it's a technique developed by Luis Pires during his PhD in Arbenz Group. The company is called Graphenical Lab. I think they are coming also here. So the technique, they're going to explain better than me, but basically they have a wax pattern. They filter a graphene oxide through the hydrophilic and hydrophobic parts of the filter, and then they can just basically transfer the pattern to whatever substrate they want, so we can achieve mass produce, and also we have very flexibility in the type of substrate that we employ, facilitating the environmentally friendliness and the multiplexing of our sensor. And finally, some thanks again to UA, super good. We have the medical team. So for example, now we have the three medical doctors, now two, because she's gonna defend her PhD next week. We have a dedicated nurse who's taking samples for us every day. We have a person in charge of the ethical and the organization compliance. And now it's the team. So basically, I'm at URV. I'm basically working in the same lab space of Kira O'Sullivan, if you know her. Her group is super good in doing aptamers of any types. They do any type of Selex, but also they have like a very multiplexed sensor. They have like a chip that can simultaneously measurement over 60 electrodes at the same time in a very little portable instruments. The other good things of her group is that she has a very senior scientist that kept the knowledge in there. So, and not only that, Vaso, she knows how to express protein, express antibody, modify it. So basically, we are really covering all of the aspects from the need to the engineering to the application. And these are our main collaborators, Andrea, Luis, and Miguel. Of course, thanks to everyone that paid me so far. And Fala. Say Vala?
Thank you for a very interesting presentation. You are ready now for the questions. So probably, do we have any question from the public or online questions? They're still sleeping, so
Yeah.
probably they need coffee.
Okay.
So I have several questions, but okay, let's start first. Which detection method do you use for the measurements?
We use square wave.
Okay, I'm asking which detection method that they use for the measurements.
Yeah, normally we use square wave voltammetry, although there have been some work that use current amperometry and other work that use impedance, but we basically use square wave voltammetry most of the time.
Okay, perfect. And for me, that's Aptamer that's fall down. It's very impressive idea. So generally, you use metal and blue, as I saw, to detect the changing in the signal. Okay, thanks. Any other question?
Yeah, I mean, there are work that use also ferrocene. We use methylene blue because we can use it in biological conditions, so we don't need any acidic medium or.
Yeah, that's nice.
My question is, like, you talk about the malaria to detect it in the blood. So do you think like we can direct directly in the blood without any purification or without any centrifugation?
Yes.
So can we go through those sensors in those area like which don't have proper hospital conditions?
Yes. Yes. Well, yes. That's the main goal. So for malaria specifically, we have two biomarkers, the LDH and HRP2. So most of the diagnostic methods use HRP2 because it's secreted by the red blood cells. So it's generally the best biomarker available. LDH is not that secreted, but it's still available in the blood. And that's why, but HRP2, as I was saying, in some area of the world has been deleted. So we got a lot of false negative results right now. And so combining HRP2 and LDH, we hope to avoid the problem of the deletion on one side. And the other side, we are basically writing the paper now. We did the biomarker discovery assay as well. And we saw that LDH, the amount of LDH is related to the severity of malaria. So we hope that by detecting both, we avoid the false negative, but also we can achieve prognostic availability of the sensors. And for using direct in the blood, yes, I mean, now we use 1% blood, but it was the first work that we did in the group. And there are many different things we can do. We could put, for example, a blood membrane on top of it, like we do lateral flow assay.
Suppression and all, yes, that's majorly like, because if it's 1%, if we use this whole blood, it coagulate like within few minutes. Because I remember like.
Yeah, well, I mean, like for example, the work that I show with the rats is done in real rats. So it's in the circulating, so it works in 100% blood. The problem is that depending on the concentration of your target, you get. So we need to work a little bit on the.
Yeah, but that's impressive, like the determination of new biomarker for that malaria, because that's the priority of need.
Yeah.
I'm basically from India, so that's like one of our own area. Like we have most of the malaria cases there.
Yeah.
Thanks for the talk. Thank you.
Thank you. Okay. Well, if you have any questions, no, there is another question.
Thank you. You mentioned that the stability in a temperature range is okay with these Aptamer sensors, but what about enzymes which are present in biological samples and can destroy Aptamers? Have you studied this type of stability?
Me personally, no, but some friends of mine did. My friends, some colleagues of mine did. I think what they saw is that, since most of the enzymes that destroy DNA, they attack it from the sides. But in this case, since we have the DNA attached on the surface of our electrodes, one side is already covered, and the other side has the methylene blue, so the enzymes cannot really attack it much. And but anyway, the stability tests show that we can get very good stability for several hours. I mean, up to 15 hours or more. And the other thing, yeah, I should have probably, but I can share the manuscript. The fact that you can have different signals at different frequencies and amplitudes allows you to correct also if there is some deterioration, you can correct it, and so you can still be able to quantify your device.
Thank you.
And we are open to collaboration.
Okay, any other questions? If not, thanks, Paolo, once again.